Okay, so let's continue to discuss auditory system. So I started last Friday for this one, and I showed you the outline of central nervous system for audition, and then mentioned there's two very important computational principles I'd like you to understand, and I'll use the examples to show this. Now, when we talk about how the brain processes sound or any other sensory stimuli, there's a very important notion that how does the brain initially convert physical energy into internal representation, and then how does the brain transform that into the presentation that eventually related to our perception. This is a process that's very complicated and long, and I will use the example here to demonstrate these two, I think, is very important principles. The three questions I'm going to continue to cover are here, and we begin with this last Friday. Now, here's the issue, here's the problem I highlighted at the end of my last lecture. For those of you who didn't quite catch up, in short, for a sentence like this, if I say today is Monday, it contains slow variations we call the syllable. Then faster variations we call the pitch, that tells the gender. And then even faster or finer information we call the speaker identity, dialogue, so far so on. If you hear someone speaking English coming from different parts of the country or from different parts of the world, the local and individual dialect show up here. Now, this is a question that engineers and psychologists and brain scientists have been wondering for a long time. That is a large time scale. I said at the beginning, end of the last lecture, that if you're an engineer, we're asked to design a computer to capture all of these variations. If you've done very well in signal processing, I think most of you are, and if I ask how you're going to design a machine, the first thing comes to your question is to look for the highest frequency or shortest time intervals for which your system has to cover. For here, if it's in a millisecond, 1, 2, 3 milliseconds, then you have design digitizer that samples at least twice as high. This is a Nyquist sampling rate. That's what you learn. But by doing so, you would oversample all of this. This is a problem like if I'm going to ask you, give another example. If I'm going to ask you to measure the distance from this wall to that wall with a precision of 1 millimeter, how would you make a ruler? How would you make a ruler? Okay. If you make a ruler very short, it will take forever. I say, well, you do this in 10 seconds. Well, you can have a major ruler that are a couple meters long, so let's say 30 seconds, 10 seconds, so that you can run a few times on there. And then that ruler has to be engraved to millimeter scale. That's how would you do. That's exactly what a sampling rate would do. But what I'm going to illustrate with the example that follow, is not what an auditory system do. This is one beautiful example showing you nervous system accomplishes particular task using strategies very different from what we have in a computer these days. So all of this start with this phenomenon called face locking, which we explained more than once now. For now, you should be able to explain to your friends, to your family, whoever with reasonable education, why auditory system, auditory nerve has face locking. This is all because of hair cell when they vibrate, push forward and backwards, only one direction channel is open, therefore you have firing. The other direction, half face, no firing. That's cause of face locking. So all the timing signals auditory system capture from the sound come from here. This is the beginning. Okay. And then we also said this property only exists, face locking exists in low frequency. As frequency increase, the ability for neurons to face locking become worse and worse, eventually disappear. This data from cats, people believe is similar for humans. And where is this? 1 kilohertz, 2, 3, 4, 5. By 5 kilohertz above, there's no more face locking. So that means if you were to plot this figure, but change the frequency to 5 kilohertz or higher, you will not see this. You will see flight. Okay. Now this is interesting because if that were the case, if a face locking is what the auditory system can use to capture the time varying component in the sound. This one goes for about 1-2 kilohertz. It becomes getting worse. Now, worse yet, this is auditory nerve, the first curve here. And that's connected to hair cell. Now, if you go to next stage where auditory nerve inputs are integrated, processed by second stage called cock nuclei, if you repeat this face locking measurement, you will get this curve. This curve shifts towards the left. That means if you look neuron here, face locking would completely disappear at 2 kilohertz. Then you will move to another stage. As we move along the pathway, this curve will keep moving leftwards, eventually about here. This is outer cortex. This is where if you have lesion, you have damage, you will not hear any sound. So that's crucial for your hearing. Face locking is about 100 hertz. Okay. And I said last time, I asked some of you what is the sampling rate of CD or MP4, MP3. That's about 40 kilohertz. And 100 kilohertz is 100 hertz is very pitiful. If you say, I told you neuron in our brain cannot follow anything faster than 100 hertz. And we're not supposed to hear music, speech, for song. So how does the brain, why brain has this? And how does brain deal with the component faster than 100 hertz at this level? Why is this? I explained last time a little bit that this shift for this curve leftwards is inevitable because integration of inputs, because of low pass filtering, all of that. Okay, and we're not going to discuss there. Now, how does brain do this? So in fact, you have already done that experiment yourself. That's the first homework I gave to you, where you are asked to hear a sequence of sound and then ask yourself when your perception transition from discrete to continuous. It turns out, if you were to play this sound to a monkey and record from neuron in monkey's auditory cortex, this is what we see here. Okay, and until now, you probably have never seen this picture. And in about a couple months, later this year, the newest textbook, Principles of Neuroscience, that's meant for medical students, will have this picture. So this will be going to a textbook in coming years. But I'm sharing that with you now. This is a very interesting observation. That is, if we recorded one neuron from outer cortex, we play the collect train. Faster, faster, right? The neuron would respond like this. Now, this is so-called data raster that we discussed in one of my earlier lectures. And each data here represents action potential. So each of this collect train was repeated 10 times. So this is a 100 millisecond interval. So there are 10 trials here. That's why I line up just for you to easy to see. Okay. Then 75, 70, so far so on, getting faster, faster. Here we go. Last Friday, I was not able to show the picture. Actually, not for good reason, because I asked you, some of you, where was the transition point for you when you did that test? It's about 40 some hertz, around 40 hertz, probably somewhere. Don't get nervous if it's not 40 hertz. Everyone is different. We're not going to penalize for your data. But roughly speaking, I've done this for many years for you, for students here, for students in China, elsewhere, for same example. Every time I twist the lump a little bit. So if you look at last year's, this was a joke, lightly. So my homeworks I give every year resemble some of the past. But if you think everything is identical, it's not. I tweak things here and there. But the lump transition for all of you statistically is somewhere between 20 hertz to 50 hertz, roughly, somewhere in the middle, right? Does that make sense? Okay, most of it. So what is that if we translate it to millisecond? So it's somewhere around 30, 40 millisecond. It's here. See, you see it's a neuron. This is not a neuron in our brain, in human brain. It's in monkey brain. So when monkey hears that collection that you hear when you did the homework, the firing began to initiate very nice face lock. And it become jitter around here, eventually completely disappear. That's 25 milliseconds is 40 hertz. This is exactly where your perception changed from discrete to continuous. And this is actually very stable. But here's the question. So this is nice. You said, this is very nice showing you. But what about the rest of the sound? This sound is this now 1 second. You hear. As you did in that homework, you hear this sound all the way through, right? You hear it. It's not discrete, but you still hear it. But look at this neuron. This neuron has no firing, nothing. So this actually puzzled the scientists for 30, 40 years until this year. Until this year. So this study was done by a former graduate student from my laboratory, Tom Snow, who was a former BME student, just like one of you. He was our BME student, and he stayed on to do PhD in my lab. In his thesis, doctoral thesis, he discovered that in the auditory cortex of monkeys, when monkey was alert, there was other half of a neuron people never saw until this work. That's the data following. Okay? Same stimuli sequence, different neuron. This neuron, you see, didn't respond much when the interval was long, when this was slow. When this become increasingly faster and faster, the neuron firing become more and more. How do we quantify this? So if I ask, how do you quantify this? Or you know this already. You would calculate the firing rate during this interval and plot here as a function of intercalary interval. That's firing rate. You see, as you move this way, firing rate increase. Okay? But importantly, you do not see time structure as you see here. There's no time structure. Stimulus has time structure. Neural firing has no time structure. That means this neuron in cortex has integrated, converts the time structure, time varying signal into firing rate. When you change the firing rate from 20, when you change the interval, interclock interval from 20 to 10 milliseconds, it would increase firing rate, therefore being able to tell that is firing rate, the clock has been changed. So these two types of neurons are two examples to demonstrate one of the computational principles I showed you. That is, this kind of firing, we call it synchronized response because it's really synchronized to stimulus. This one is called a non-synchronized, but it's because it does not synchronize this. But what if this response is something we can categorize as isomorphic or fistful? Because without seeing this, if I give you this, say what's in stimulus, you will tell, because this literally captures this. This kind of response, isomorphic, in this case, is almost like another example. If I put my hand in a Xerox machine, I push a button, what come out is what? Exactly my hand. Okay. That means if you project the hand, if you look here, if you look at my retina, this hand is identically projecting my retina, just like a Xerox machine. This is called a fistful transformation, okay, or isomorphic. This is non-isomorphic now, because if I just give you this, you could not tell what's in stimulus until you do other things. Okay, this is considered transformation in our brain now. So now you know, when you hear click trend at 60, 70, 80 hertz, as you play it, you hear them, that continues in your brain, the neuron like this, firing continuously in response to the click trend. So that's correlation. This is one example. And so that's the principle. This example demonstrates that the central nervous system transforms the isomorphic or fistful representation of a sensory stimuli established in the peripheral to non-isomorphic representation in the brain. This is one example, very simple example. I'd like you to spend some time really understand what does it mean and be able to generalize this to other examples if you are given. Okay. Now, these two populations of neurons that we talk about, I gave you one example each. And you need a lot of neurons to do this, so that if you look at 100 neurons in auditory cortex, half of them are synchronized, and you can quantify vector strengths, as you learned by now. And you can say vector strength goes down as interval becomes shorter or sounds become faster. This is a blue line. Now this population, you can see, we call it temporal coding because it can encode the time structure explicitly. Whereas this population, the red one, we call non-synchronized population, there are more and more of them. Firing rate increase as interval becomes shorter. And they encode the sound with a firing rate, we call it implicitly. And the border of these two populations is where your perception boundary of discreteness. It turns out this boundary is very similar to voice onset time of speech. So when you say ba and pa, all the differ is about 30 millisecond noise. When you say ba, if you feel it, ba goes out right away. When you say pa. If you do it now, you notice you actually hold back a little bit. Pa. You hold it back about 30, 40 milliseconds. There's a little bit of noise here. All you do is that's how you do it. That's a very important parameter called voice onset of time speech. That's also on the boundary here. Okay. Now, what we see this curve here, another notion is that this is in the primary auditory cortex. But where does it come from? If we were to do this experiment, exact experiment in auditory nerve, what would we see? You will see auditory nerve data. If we were to play this sound, sequence of sound from 10 hertz all the way to 300 hertz, and record it in auditory nerve, what would you see? You know the answer now. What kind of response would you see? Okay, think about this. I'll leave it. Think about it. If I give you ask a question, you should know that now because you know what an auditory nerve is. Anyone want to offer? Guess? Okay, I'll leave that. Let you think about it. Think about it. Okay, but this is what I would say in the cortex. What about the stage right before the cortex? That's the thalamus, right? And here is actually thalamus. So this red line, two lines here are in the cortex. This is in the thalamus, two population. And notice the thalamus, the synchronization, this curve, it goes to more towards the left, a shorter interval, that means faster. And the cortex is here, okay? Thalamus here. That means the thalamus can face lock to, just like we said earlier, a faster synchronization. But it's still not fast enough to the auditory nerve. Auditory nerve is somewhere here. It's 1 millisecond. And therefore, thalamus, we call MGB here, medial genetic body is the auditory part of the thalamus. Also have firing rate here. Also have this combined representation, but in cortex is shifted. So the notion here is following. You start with auditory nerve. The phase locking upper limit describes that curve that goes down all the way to 4, 5 kilohertz. That is all the phase locking auditory nervous system would have. Anything higher than that, there is no phase locking, there is no timing information. As you move along the pathways, phase locking upper end of that become lower, 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 eventually down to about 100 hertz. At the same time, another population of neuron, we call the non-synchronized neuron, began to emerge. As the phase locking goes down, that population gets emerge. Eventually, these two populations together encodes what we hear, the whole range of sound in a sentence like, "This is Monday." That's meaning spoken by male speaker, and it's myself. Okay. All that information is encoded by these two populations. So in other words, that if I will go back to that example of measuring distance between here and here require 1 millisecond precision, the nervous system built a scale, one ruler that's just very long, 3 meters long, and just run this whole thing for the big distance, and then use another finer, different ruler for that little bit left to do. Okay, so that's the first notion of this. How does the auditory system represent time varying signals? And this is important not for audition, but for all other systems as well. The second question I'm going to discuss today is how does the auditory system represent a spatial location? This is something that you involve every day. For example, if I'm letting you hear my voice here, if I'm standing here and just suddenly completely turn off this lights here, become darkless. And people still have no problem knowing where I am. People still on this side knows I'm to your right, here to your left, and here in the middle. Okay, so we do that all the time. In fact, this is one thing that we use every single moment. So how do we tell sound come from which side? Well, it's simple physics. If this somebody's head is nose, 2 ears, if the sound comes from the right end, okay, it will reach your right ear first, left ear a little bit later. Because our head has particular size, from simple physics, you can calculate. There's a time difference, delta t, we call it interoral time difference. It's in the order of several microseconds, very tiny. But it's big enough for auditory system to use. So if the sound comes from the middle, delta t will be 0. Okay? Now, in the same vein, because this path, sound from the right side to the right ear is shorter, it's a little bit slightly louder than sound here because sound attenuates as it propagates through the air. So there's a difference called delta I, interaural level difference. And physically, in terms of physics or mathematics, these two information are equivalent. So you need one of these to calculate sound from left and right. But why we need two? We'll get to that point in a few minutes. Okay. Now, how about the sound come from front? When you are me and in a straight line, your both ear receive the identical timing and intensity from sound. Okay. So it turns out the way we tell sound come in front of us in the middle line, whether it's in front of us, below, and above, or even behind us, is because of their ear. This is one thing I mentioned early on when I talk about the ear. Our ear is not for cosmetic function. It's actually for localization. So our outer ear pinna here, serves as an acoustic filter. It creates a notch. That is, if you play a white noise here, record sound here, then what you record here, white noise has a flat envelope. This is no longer flat, it has a notch. This notch changes as the location of sounds changes. So this is how we tell sound from front and back. So that's why if you lose your outer ear, or if you do something just artificially block it, you will find the location, your ability to localize the sound along this line impaired. So these are three parameters that allow us to determine the location of a sound anywhere in this space, okay, without a vision. So how does the auditory system encode here? And here's some kind of psychophysical data, behavior data, show you how well we do. And people have studied this for half a century. So if you put somebody in the middle of a testing room and put a speaker at various locations, ask someone identify where the sound comes from and measure how good the person does. So in front of a person, if you play a brief sound and each location and its standard deviation, the error is used to here. You see, we do pretty well. You know, our error is within a few degrees. But if you look on the side, we do a little bit poorly. And look on the back, we do a lot of poor. So sound from front, we do very well. And we do a little worse on here. And of course, all the performance improve. If you make sound continuous, then you have brain has more time to calculate. But how does the brain calculate here? So we don't have time to go through all detail of sound localization. This is a very complex subject. But I want to highlight just two key elements. How does the brain extract interaural time and intensity difference? So let's go back to, oops. Okay, let's go back to the anatomy here again. Okay, so here's the inner hair cell, auditory nerve fibers. It come in here, cochlear nuclei is here. Then it goes through this structure we call MSO, LSO, MSO. That take inputs from both ends. Now we have both ends. Okay, so this is a structure part here, also part here, called a medial superior olive. Don't worry about the name. I'm not going to ask you if you're blank. But there are two parts, one like a middle part, a lateral part, superior olive. And that is the name I spelled here. And each of them calculate interaural time difference and frequency difference. Okay. Now how does it do this? So let's look at MSO. So MSO is this structure here. Okay. It receives inputs, excitatory inputs from same side, ipsilateral, and opposite side, contralateral. Is contralateral. Okay. So here's how does it work? Okay. Suppose there are group of neurons in the MSO. It get connected to the cochlear on one side, and the cochlear on other side. Okay. And you have sound come from loudspeaker to the left. Okay. So when they make a sound, it travels to left ear first. It takes a little longer, a little bit later, travel to right ear. Once, suppose this is tone. Suppose this is tone. It comes to left ear, and with everything we've discussed so far, will generate action potential, and running through here. And this will generate action potential that running through here. This Thursday, Friday, we're going to talk about conduction velocity. That's everything applied to this propagation. Now, Now we come to the neuron here. So a neuron in this MSO has a particular property. It does temporal integration, but with a very short integration window. Okay, this is a concept again, we're going to test it in the lab. You're going to do with me on Thursday and Friday. The fourth experiment in your lab, temporal integration. So the temporal integration window is so small that you almost require two spikes arrive at the same time so that it can bring the neuron into over threshold firing. Okay, so we call this coincidence detection. Okay, so now if you look at the action potential come from here, in order for action potential from this end to meet with action potential here, it has to be in this neuron, because it arrives here first, it begins to run, it takes longer to run here, arrive here later, begin to run, takes shorter to run here. So this neuron would receive action potentials produced, evoked by this sound from this ear and this ear at the same time. Okay. Other neurons would also receive action potential from both ears, but because of the mismatch of distance and time, they will not arrive at the same time, so nothing happened. Remember, the key for this neuron is that it has a very short temporal integration window. We call it coincidence detector. Action potential has arrived at the same time, so therefore this neuron would discharge when sound comes from here. Now, what about if we change the location? Sound comes from the right end. For the same token, same argument, this neuron would fire. So therefore, you can see here, location along this space, different location, will be encoded by each of this neuron. This is called coincidence detection that you calculate basically delta t. Okay. Now, here's a question. In order to this happen, here's a question now, I'm throwing back to you now, think about it. If I put this in the exam question, what kind of tone I put in here that will make it work? Can I broadcast any sound from this speaker, sound with any frequency that will make this circuit work? Can I? Yes or no? In order for this to happen, or let me give you one more hint. In order for this neuron to detect the near simultaneous arrival of spike from here, what kind of a property of action potential that is expected on here? Can there be any action potential? Now, by now, you have done analyzing action potential, you have done simulations. The exam homework I gave you last Friday is asked you to simulate a Poisson spike train or modified Poisson. Okay, let's use that example. If action potential here running along this axon here is Poisson process, and the same one here, Poisson process, can this work? Can this work? Anyone guess? If it can't, why? Now remember, I just said earlier, this neuron, what's special here is that it has a very short temporal integration window. That doesn't mean two spikes coming very close in time, near simultaneously, for this neuron to fire. If you have a Poisson process here, what is the probability that two events happen same time, high or low? Very low. Yeah, some of them not, very low. So literally so low that you cannot expect to happen again. Okay. Therefore, if you have an ideal Poisson process as you model here and here, you can simulate this. You can actually run a short code. It will never happen. Then what kind of action potential train that this can happen? This is related to the question I asked a moment ago, that what kind of sound? Because this action potential is generated by sound you stimulate here. What type of this is tone? What a frequency of tone that can work? Anyone can say? So if Poisson process doesn't work, what else we need here? Phase locking. You need a phase locking. You need a phase locking, so spike train on either end the phase lock, so they can meet once, again. Okay, they have meet many times. You need phase locking. Now, if you need a phase locking, then the rest is straightforward now. Then you know what kind of a tone that is. This mechanism only work when there's a phase locking outer nerve. Because the phase locking is a very refined timing, therefore, this allows you to measure simultaneous arrival up to down to microseconds. Okay. Now then, here's a question. I just mentioned earlier, phase locking goes down, completely disappear about 4-5 kilohertz. But yet, if I play a song here 10 kilohertz, you have no problem till left and right. Then what do? You go to a second mechanism called the interval intensity difference. So this two cues between our 2 ears, time intensity, mathematically they are identical. But time difference can only be detected through the phase locking mechanism of 2 ears at a low frequency. At high frequency, the nervous system has no more phase locking, no timing, and it has to use interval intensity difference. That's why there are secrets here. Now, this is just a further, in the interest of time, I'll let you read it here in your handout. Let's just show you the mechanism we just talked about here is also called a delayed line mechanism. And it also turns out this mechanism, as you saw in last slides here, this is in every textbook. They're only half correct. So that's why in this class, we don't follow textbook down to every page. This is correct, but only correct, it turns out, if it's birds. For humans, mammals, monkeys, this does not work at all. Okay. But for the textbook, we still use it. What happens? So this is our phase locking I mentioned to you, that govern where interval time difference. So for mammals, there's still interval time difference calculation, but not through delay line, through the combination of excitatory inhibitory ones here. And the detail of this, I'll let you read it. But the general concept is the same. Only time work at low frequency. This is comparison between two models, between birds, we just talked about, and mammals. Okay, I'll let you to read it here. And this is something that you should pay some attention. Now, what about the interaural intensity difference? Interaural intensity difference is calculated in another structure nearby called the LSO, lateral superior olive, where the neuron here receive excitatory inputs from one ear and inhibitory now, inhibitory from another ear. The MSO neuron is different. This is the LSO neuron. So the inputs from other ear through interneuron become inhibitory. Now, the neuron here, temporal integration window, in contrast to MSO, is long. They have long integration window. They don't care two spikes arrive about the same time, but they care within the window how many spike is here. This is the worst concept you're going to test in experiment number 4 on Thursday or Friday. So therefore, whether this neuron fire or not is proportional to net excitation, right? So that means if you have more spike from left end, there's more excitation than fewer spike from the right, from less excitation. If sound comes from the middle, both of them have the same. And why we say sound comes from left, have more spike here? Because if a sound comes from left, arrive here first, is a little louder than here. The fire rate here in outer nerve is proportional to sound level. There was a couple of slides in last week. I flashed through very quickly. My previous slides. The outer nerve firing rate is proportional to sound level. That is showing about here. So therefore, neuron here can calculate the difference between fire rate and the consequence is difference between the sound level. That is the interlevel difference. Therefore, tell sound from left and right. So in summary, to tell sound from left and right, if it's a low frequency, auditory system use interaural time difference. If it's a high frequency, use interaural intensity difference. If you're in the middle, auditory system detects a notch in the ear. Okay. And that's another summary of this. So in last about 15 minutes, I will explain the last one, perhaps probably most interesting one among a number of things to do. That is, how does the auditory system process pitch and harmonics? So I raised this question earlier. Yes?
Sorry, with the interaural intensity difference,
yeah.
If you have an intense enough stimulus, can you blow out that mechanism and just have something too loud to localize?
Sure, good question, good question. That is, if you have a stimulus so loud that is saturated neuron, then you won't be able to. It's a very good question. Thank you for asking this, because in that slide I went through in my last lecture, we talked about when sound become louder, firing rate increase, eventually flat, become saturated. That's very loud. When that's loud, auditory nerve cannot tell whether sound is becoming ever louder. Yes, this can only work when intensity at the level when change, sound firing rate change. Yeah, good question. Any other questions? Okay, so the last subject I want to cover today is regarding how the auditory system processes elements about music. This is something I mentioned to you earlier, and the music and the speech are related. Now, here is diagram showing you in humans how we produce speech or sing a song. Okay, so here is the side view of our speech production apparatus. The lungs here, so air comes from air here, and there's a structure called vocal fold. It's like a memory, half open. There's a gap here. So when the air punches through here, flows through here, it becomes vibrate. That vibration is what we call a pitch. So for all the males, if you put your finger here, there's a bump here. Okay, female doesn't have pump, but it's the same thing. You put your finger here, you say, ah, you can feel vibration. If you whisper, say, hello, today, there's no vibration. So only when you vibrate, when you push air through here, the vibration here gives you a pitch. Okay, so that sound comes through a generated pulse with a pitch. Then depends how we place our lips, tongues, how we configure our mouth. Then this serves as a filter. Together, you get this signal that we talked about last time. Okay, this vowel when you say okay, and the pitch that generated by vibration here is the frequency of a first component. That is the periodicity we show here. Because it's a harmonic, second one is twice, third one is 3 times. That's spectrum of this. So when you hear my voice, then you know it's a male voice. It's because you compute the pitch of my voice is somewhere here. But another thing interesting in a moment we're going to tell you is that you have been fooled by engineers for years. Ever since Bell Lab scientists figured out, it turns out that when we hear, if my voice is pulsed here, that's about 120 hertz. That's a typical male voice. But it turns out, for years, engineers, when they transmitted this sound into land phone back then, now cell phone now, they never transmitted anything below about 400 or 500 hertz. It just filtered out. Why? Because you want to save bandwidth. Bandwidth is money. The narrow bandwidth, the more cores you can pack in the copper wires now, and the channel now. But whenever you pick up a phone, either land phone or cell phone, you have never had a problem tell whether it's male and female, right? Why is that? That's because in the absence of this real pitch, our auditory system computes the pitch from the interval here, because all of them are integer multiple of this. Therefore, the interval between any of the 2 components is equal to the pitch. This is how pitch is used. Okay, and I'll give you the example already in tonal language. This is crucial, and English is a partnership. Q is also useful. And here is the experiment to demonstrate that amazing ability. This is something that is probably most interesting in audition. That if I ask you to do this experiment, on 1 year, I give you a pure tone. I play one pure tone 1 year, but give you a knob, you can change the frequency, dial upside down. And the other year, I give you, let's say, 4 tones at 2, 3, 4, 500 hertz. You hear that? It's like, just like a piano play. Then I say, dial your left dial until you feel they're too match. More likely or not, your dial will stop at 100 hertz. That's pitch. Okay, pitch of 100 hertz, pure tone, and harmonic complex. This is the phenomenon we call Meson fundamental harmonics. Because in the absence of a real fundamental, real pitch, physical pitch, our auditory system can compute or extract the pitch from here. Okay, that's where Bell Lab scientists found out half a century ago. Now, if you move this 4 component to higher frequency, 5, 6, 7, 8, 9,000, you still hear 100 because the interval here is 100. Until you enlarge this interval to 250, you hear 250. This is called pitch of missing fundamental. We use this every day. Every day, whenever we make a phone call, you use this. Okay, even though most people don't aware. But how does brain process this? So the brain process this is similar to maximum or brain process this that I showed you. That you figure out there's a dark by compute this. There's no dark. So this is the example of a first computation problem. I said earlier that the central nervous system can compute or extract information that does not exist physically. For example, this dark does not really exist. If I give you an example, give you the question as the exam asks you, is there a dark or not? You insist there's no dark. I cannot take point away because there is no dark. You extract computed dark here. So it also turns out that this harmonic pattern that we as human beings, we actually prefer harmonic. This is a test that they did on the undergraduate students in University of Minnesota, where they gave a student to listen to consonant or dissonant, ask a student to rate which one is more pleasant. By and large, undergraduate students, I think you too, would rate the harmonically based consonant more pleasant than dissonant. Okay. So, but we are not the only species who have harmonic in their speech. There is vocalization from echolocating bats. You see harmonics. And here's a vocalization from monkeys, my laboratory study called marmoset. And you have harmonics everywhere. So harmonics are everywhere. Okay. So how does it bring computer pitch? So in cochlear, which by now you know this is a frequency organized. We said it's like you have piano keyboard here. And you press one key, you have one excitation pattern. You press another key, have another excitation pattern. If you play a couple of keys together, you have multiple patterns. But cochlear does not compute the pitch. Okay, it does not. It just captures all the energy. And if you look auditory nerve that are connected here, so for example, if we give this sound, this vowel is like you play couple of piano keys at the same time, some louder than some heavier, some lighter. What does auditory nerve do? Well, we'll look at this last time. Auditory nerve will show this function like this. It shows the peaks here that corresponds to format. But remember, there's a harmonic structure in this vowel, and you do not see harmonic structure in auditory nerve. Okay, so where do we see this auditory nerve harmonic structure? So let's turn out this is represented in a cortex, many-stage letter. And here's an experiment done by graduate students who used to be TA of this class, Dan Bender. He's professor now in University College London. This is from his doctoral thesis. So here's this experiment that Dan did to discover a pitch neuron. We know it now. And if you take a neuron, we call it pitch neuron. If you play the pure tone with the changing frequency, the neuron will respond when tone is in the center of a receiver field. You know this now, okay? But if you play harmonic complex tone, I'm sorry, this is non-pitch neuron. This is auditory neuron with tuning. When you change the frequency, when frequency in the center of a receiver field, this power-based frequency, we're firing, otherwise no firing. And if you play harmonic complex tone like here, this is, as we just said, if you were to listen to this, you will hear pitch, same pitch for all of them because this interval is the same. They're all missing fundamental. The first one you want to respond to because this one right in the receiver field, the rest doesn't respond. So this is what a standard auditory neuron would do. What does a pitch neuron do? Here's what a pitch neuron do. You give a tone, has response, but you play harmonic complex, response to everyone, as if this one extract the pitch from there. And here's more data. I will let you to read it, and what's the controls, and where is the pitch located? Where in our brain? I take it down the point. It's actually, this is in the monkey brain, in a very small region, in a low frequency range of auditory cortex. Remember, auditory cortex, we said that last week, is organized by frequency, from high frequency to low frequency. They're here. Now, it turns out, we know now, so that's where it shows pitch center is. Okay, it turns out we know now, from a number of studies by others, that in humans, all of us, we have a part of our brain, we call it pitch center. In the low frequency corner of our brain, we have neurons there that extract pitch generated by our voice. And that center was also found in marmoset monkeys. This is interesting because some of you would wonder now, hold on. Dr. Wang just told me all the things about Chinese, English, music. Then marmoset, monkeys? Why monkey has anything to do with all of this? Monkey doesn't listen to music. Monkey doesn't speak Chinese or no English. Okay. Where's the pitch center? Pitch center is here. So the reason, this is interesting because it has everything to do with music now. So this is a harmonic pattern of music, spectrum of music. You saw here. And in the auditory system from the beginning, so I'm going to finish 1-2 slides, or I'll tell you end of story. The neurons only tune to one frequency as you've seen. As you move on a higher level, neuron begin to tune to mod 2 frequency, like shown here. Okay, I'm going to skip this one. And then when you move into cortex, here's the most recent data showing you what the brain has to do with music. We have a neuron like this, we call it harmonic temporal neuron. If you give one frequency, has a little bit response. If you give 2 frequency, that is, have one frequency here, then I change it to second frequency. When these 2 frequencies are harmonically related, okay, you have a little more frequency. Then if we give many frequencies, so there is about 10 frequency, like 10 frequency, give same time, then increases the interval. So the interval between each component is larger and larger. You see, it is 2 frequency, that's the signal. 10 frequencies together respond very heavily. This is like you play one piano chord to this neuron doesn't care. You play 2 keys, they fire a little bit. You play 10 keys, boom, neurons fire, right? So now the fact that the neuron in the brain detect this pattern like this have a lot of implications because it turns out, okay, these neurons that detect the harmonic pattern are everywhere in our auditory cortex. Pitch neuron is in a small region. It's very special. And it also turns out we have shown the neuron sense data will come from marmoset monkey. Now we know this is actually marmoset, even though they don't speak Chinese, they don't go to karaoke hall to listen to music. They actually perceive pitch if you test them in a very much the same way we perceive it. So this marmoset now, you can know, if you give them 2 sounds slightly different pitch, they were able to derive them as well. But then the question is, why? Why we have this? This is my last slide. Where's marmoset? Marmoset is here. There's humans. This is a primate evolutionary tree. It's about 30 million years ago. There is a bifurcation. Monkeys in the New World, that is South American, here, and humans here. The fact that monkeys bifurcated so far from us have a neuron in the brain that encodes pitch and harmonics suggests that our ability, perhaps our reason to need music, has a non-evolutionary root. It is our brain that is structured to receive music, that humans create music to satisfy that need. All right? So they work like this will eventually tell us the music, the importance of music to the brain and the importance of music to the rest of the world. So there's one important hypothesis that is because the harmonics is everywhere in our environment. Now it goes to the question, why then human's brain has harmonic? That's because when we evolve, when we grow up, we hear harmonics every day. That harmonics perhaps shapes the way our brain is what it is now. And that's why we probably think particular music listen better. And here are suggested readings, and some of the details I gone through, and you should read it. And I made some changes to these slides. I'll give another version to post, and the TAs will help you to fill in some blanks. All right, so I'll see you in a couple of weeks.